Today's mini lecture is about gene mutations. You should already know how DNA replicates. You should also know how RNA transcribes and translates in a specific portion of DNA, otherwise known as genes, into proteins that have a specific function in a cell. This mini lecture will address two categories of gene mutations. These are mutations that occur within a specific gene sequence. First, we will talk about point mutations, a mutation that changes a single nucleotide in a gene sequence. Then we will talk about frameshift mutations, a mutation that results in a change in every codon in the gene sequence past the mutation. Things to think about as we go. You need to think about what happens at the location of the mutation. You need to think about what codons are affected and how. And you also need to think about what is the resulting change in protein synthesis. Sometimes mutations have no effect on protein synthesis, and sometimes it can result in the wrong protein or no protein being made at all. We will use an example, this sentence example, as we go. So we know that codons are made up of a sequence of 3 nucleotide bases. So we can think about the DNA sequence or a gene as a sentence that is made up entirely of 3-letter words. So we're going to look at this sentence, the cat ate the rat, 3 different times. And the reason why we're using this sentence is because 1, the sentence makes sense, and 2, each of those words are made up of 3 letters. So you'll see this sentence again and again during this little mini lecture. Okay, let's start with point mutations. So point mutations occur at a single location or point in a gene sequence, meaning a single nucleotide is changed. And this affects a single codon, which will affect a single amino acid. In this example, we actually have the gene sequence, and so these are the DNA bases. And you can see that a single base in the DNA has been replaced. Instead of the normal gene codes for an A, we have it coded as AG. And so what would happen is that you would end up with a change in the gene sequence when it was transcribed and then also when it is translated. So remember, a single codon is affected, and we know that in some cases, several codons translate to the same amino acid. So the result could be, if the same amino acid is coded, translated, then there will be no change in the protein synthesis and life goes on as normal within the cell. If a different or no amino acid is translated, well then, yes, we will have a change in protein synthesis. So let's look at our sentence, for example. So if the original normal protein was the cat ate the rat, if say we had a substitution towards the end of the gene sequence and we have the single change, the R has been substituted and therefore it's been translated into an M. So we have the cat ate the mat. So the first sentence makes sense. The second sentence, not so much. All right. Point substitution is pretty easy. All right, let's move on to frameshift, which is a little bit different. When we talk about frameshift mutations, we're talking about a shift or a move in the reading frame. So the reading frame is basically how we read those codons. We look at codon to codon during translation, and we translate that RNA using 3 nucleotides at a time. All right. So in a frameshift mutation, we're going to have a shift to the left or to the right. And typically this is catastrophic, even though it sounds like it shouldn't be. It's just one off, but everything changes from the location of the mutation onward to the end of the gene. So here's what I'm going to tell you to do. You need to find the mutation. That's where the first change occurs. And then you need to look after and compare it to the normal sequence. So figure out which way did it shift, to the left or to the right. There are two kinds, insertion and deletion. We are going to start with insertion. So in assertion, it's just like what it is. A base is added to the code. So you end up somewhere in the gene sequence with this extra nucleotide. So if you compare the top, which is the normal sequence, the first codon is fine. We have an addition in the second codon. And as you can see, everything shifts one to the right. So we should have GGC, but if you look at the bottom, we have GGG. But if you compare the rest of it, you can see that it is shifted one to the right. So it should be GGC. Where is this original C? It's on the next codon. So here's the C. Then you can look, the start of the normal codon is a U, but here it is on the bottom, it's in the middle. A. Look at the last codon in this example, the A. So you can see if you compare the top, the normal, to the bottom, where the mutation has occurred, you can see that everything has shifted one to the right. And I should point out in this example, we're looking at sort of the resulting transcription versus translation, because all of this is RNA. So in this example, going back to our sentence, the cat ate the rat, that's the normal code. That makes sense. The ka tat f era t makes no sense whatsoever. So you can see how it's catastrophic. Who knows if this codes for a protein, but it looks like nonsense to me. And that's sometimes what happens in your gene sequence. You get these nonsensical things and it doesn't code for anything, and that can be a problem. So insertions shifts to the right. So let's take a look at the other direction. Deletion. So a deletion, something gets deleted. So a single base is removed from the code. Everything shifts to the left. So you can do the same comparison to the top to the bottom and see that things have shifted to the left. So we end up with the cat ate the rat. That's normal. If you took away the C of the cat, we got the at, her, at. Again, nonsensical, makes no sense, catastrophic. So a quick review. Here they all are in comparison. So you have the normal DNA, and then you have substitution, point mutation, and then the insertion and the deletion in the Frameshift examples. So what you would be expected to do on an assessment, you should be able to identify which type of mutation has occurred if you are given the normal DNA sequence, and if you are given the protein, and be able to compare the two things with a mutated strand. So you should be able to identify what type of mutation has occurred, and you should be able to identify the resulting effect on protein synthesis. And that is about it.